Now let's continue with our program. I'm excited to introduce our next speaker and a collaborator of mine, Dr. Brian Wiltgen. Dr. Wiltgen is a professor of psychology and a core faculty member at the Center for Neuroscience. He studies cellular and molecular mechanisms of memory formation in the hippocampus and neocortex. And his research focuses on the neural processes by which new memories are encoded, stabilized, and maintained. Is this better? Okay. So his research focuses on the neural processes by which new memories are encoded, stabilized, and maintained over time. To examine these issues, he uses rodent models and combines behavioral analysis with pharmacology, electrophysiology, calcium imaging, optogenetic, and chemogenetic techniques. Today, he will share his research on how dopamine shapes our memories of important events. Please join me in welcoming my friend and colleague, Dr. Brian Wiltgen.
Can everybody hear back there? Okay, good. So I apologize, the mask isn't helping. I had a tooth extracted, so I'm kind of embarrassed. But the last talk is causing me to face my fear and my embarrassment. So please just look at that face that has two teeth. Focus on the screen, not me. So I'm a professor in psychology, and my laboratory is located in the Center for Neuroscience. I moved here in 2012, and one of the main reasons was for all of the excellent faculty that we have who study memory and plasticity. So you've seen some of them today. There are many, many, many more on campus, and that makes it a great place for both me and the students in my lab. Okay, so I've been studying memory since I was an undergraduate at the University of Iowa. And the reason is that I find memory fascinating. So all of the experiences that have happened to us in our lives, the ones we can remember, are all somehow stored in our brain. My son once said to me, Dad, I feel like I have a time machine in my head. And I said, you do, it's your brain. That's where all of the information is. So these types of memories that you see here are called episodic memories. So these are the types of memories that people mainly think of when they think of memory. These are personal, autobiographical experiences that have happened to us personally. And so I'll focus on this today, or animal model of it. And so in my lab and also in the field, there are some major questions that we want to answer. So first, and this is what I'll start with, where are memories stored in the brain? And then how are memories stored in the brain? What are the mechanisms? How do the neurons and all of the cells we have in our brain do it? And then finally, the thing I'll focus on today, which is why are some experiences remembered while others are forgotten? And what I'm going to show you is that dopamine is one of the essential factors for determining whether or not a memory is forgotten or remembered for a long period of time. But first, a little history. So the search for the physical location of memory in the brain began in earnest way back in the 1920s with Carl Lashley, who was a famous psychologist at Harvard who was studying the brain. And he wanted to know if he could find the location of memory. So he decided to systematically damage parts of the rat brain to see if he could find an area that caused them to forget. And so he called his quest the search for the engram. Now, engram is, it's an older term, although it's become popular once again. And it simply means the physical basis of memory. So Lashley wanted to know if memories are stored in specific places in the brain. In particular, he was focused mainly on the cortex. So to answer this question, he trained rats on various learning tasks. Then he would lesion some part of the brain. Then he would bring the animals back and give them a test to see if they still remembered. So it looked something like this. So Lashley might train a rat to navigate a maze very effectively. And then once it had learned this, he would damage or make knife cuts to different parts of the brain. Here you're looking down at the rat brain, so this is the cortex. And in different groups, he would damage different parts of the brain and bring them back to see if they could remember. And what he found was nothing. He could not reliably find any part of the brain, the rat brain or cortex, that when he removed it, produced amnesia. So his quest was a failure, and you can really feel his disappointment when you read the summary of this paper right here. I'll read it to you. So this series of experiments has yielded a good bit of information about what and where the memory trace is not. It has discovered nothing directly of the real nature of the engram. I just imagine him like in his study late at night with a bottle of scotch. He's just like, keep writing. I sometimes feel in reviewing the evidence on the localization of the memory trace that the necessary conclusion is that learning just is not possible. Swig. It is difficult to conceive of a mechanism which can satisfy the conditions set for it. Nonetheless, in spite of such evidence against it, learning does sometimes occur. So no localization whatsoever. He concluded that memories were actually stored everywhere in the cortex. That's the only way to explain his results. And that was the prominent view for a while. That is until patient HM, which many of you may have heard about already. So HM had a bike accident when he was young, and he developed seizures. And over time, they became worse and worse, and they got so bad that the parents decided to have an experimental brain operation. So in this brain operation, what they did was to remove quite a large portion of HM's medial temporal lobe, and that's because that's where they found the seizures were originating. And so let's see if I can get the mouse up here. So this area right here, that's what they removed in HM's brain, and then you can see what was there in a normal patient's brain on the right. And so this part of the brain contained an area you heard about earlier called the hippocampus. Now, the good news is that HM seizures got better. The bad news is that it was immediately clear that something terrible had happened to his memory. He completely lost the ability to form new episodic memories or retrieve even some older episodic memories from his life. So experiences like you saw on the video earlier, those were lost in HM. He said he feels like he constantly wakes up from a dream. So interestingly, his language, reasoning ability, perception, and even some types of learning and memory, like skill learning, were completely fine. So it seemed like he had a relatively pure deficit in episodic memory that was caused by damage to a specific part of the brain. And later studies would tell us that it's the hippocampus that's particularly important for this type of memory. Okay, so now here you can see the hippocampus in humans, monkeys, and rodents in red. And we now know that the hippocampus is important for memory in all of these species. Now, I work with rodents in my lab, and unfortunately, we can't ask them what happened to them during the day and have them tell us what memories they formed or have not formed. So we've developed a number of learning tasks in the lab that are very sensitive to damage in the hippocampus, and I'll show you one of those in a second. So at this point, some of you may be thinking, why didn't Lashley discover the hippocampus? Well, this is unfortunate for Lashley, but at that time it was believed that higher cognitive functions, which included learning and memory, were all done in the outermost part of our brain, the neocortex. So that's the gray part. You can see we have a lot of it. The monkey gray part, and then also not quite as complex in the rodent, but they still have neocortex. And the hippocampus sits just under the neocortex. Oh, so close. So close to being famous, he's now infamous. Too bad. Okay. So we form lots of episodic memories in our life. You'll form a bunch of them today. But on a sort of daily basis, most of them are forgotten. All right, so for example, many of us might take the same route to work every day, but we don't remember most of those trips, right? Maybe for a day or two, and then they're off into the ether. That is, unless something salient or unexpected happens during that commute. Oh, sorry, Jennifer, I didn't warm him. I wanted to get a bigger reaction. She's fine. She's totally fine. Completely lucky. This is like everyone in Davis's worst nightmare, getting hit by a car. But if this happened on your way to work, you would remember that commute for a very long period of time. Why? Well, there's a number of reasons. I'm just going to mention a couple. First, salient events are better remembered because they're arousing and emotional. You could hear the whole audience respond to the clip, and you weren't even seeing it live. You just watched a video, and you had an emotional response to it, and that can enhance memory. In addition, salient events induce the release of neurochemicals that promote neuroplasticity and memory formation in the hippocampus, but other regions as well. So this kind of fits in with our focus on circuits, and you saw this at the beginning with the introduction from Tina Kim. The hippocampus doesn't work in isolation. In order to do its job, it has to talk to other brain regions. And in this case, there are some brain regions, like the locus coeruleus, that's hard to say, so let's call it LC from now on. The LC responds to salient events like the one you saw in the video. And when it encounters a salient event, it then sends a signal to the hippocampus that says, remember this. Don't forget this, it's important. And the signal that it sends is neurochemical in nature. It releases noradrenaline and dopamine into the hippocampus, which, as it says there, enhances plasticity and allows the hippocampus to form a memory that will now last a long period of time. And today I'm going to focus exclusively on dopamine. So I'm sure everyone's heard of dopamine at some point. You heard it in one of the earlier talks from Karen. And most of the time, people don't think about memory formation or long-term memory formation in dopamine. The first thing they usually think about is reward, things that we like, and that's true. In certain parts of the brain, dopamine release indicates that something rewarding has happened. But dopamine does just more than just that. In other parts of the brain, as you may know, it's important for motor movements. So this is evident from diseases that affect dopamine neurons like Parkinson's, and that has shown us that fine motor movements, handwriting, walking, gait, those things are also shaped by dopamine and are impaired when dopaminergic neurons die. So this tells us that dopamine serves different functions in different brain regions. And today I'm going to show you that dopamine has another function, which is to enhance long-term memories in the hippocampus. Water. Okay, so how do we get rats and mice to form long-lasting memories like the one you saw in the video? Well, there's one thing that really gets a rodent's blood pumping. Jasnu alluded to it earlier, a cat or any other predator that wants to eat them. So if a rat had an experience like this and it survived, it would remember this for a long period of time, and in the future it would probably avoid this particular area. So as Jasnu showed you earlier in the lab, we can model this type of aversive experience in a very simple way. We play an auditory cue to them, something very innocuous. They can hear it, but they really don't care about it. And if that's all we did, then they would forget about this cue very quickly. However, if something aversive happened, in this case a foot shock, which in the laboratory we think mimics an attack, so they kind of feel like something's biting their feet and they get kind of scared and startled by it. If we do that, then they'll remember this auditory cue for a very long period of time, months, and in some studies as long as a year, which is most of their life. So how can we tell they remember it? Jasnu showed you this. When we play the auditory cue, they exhibit fear responses, and we can measure and quantify those in the lab and use them to index memory. So the question is dopamine necessary for this kind of learning, or this type of memory? To test that, we use newly developed or newly engineered fluorescent sensors that can detect the release of dopamine in real time. So these are relatively new, and my lab was one of the first to use it in the hippocampus during aversive learning. So as you can see, we put the sensor into the hippocampus. We then implant an optical fiber just above, and that can detect the changes in fluorescence, which then indicates to us that dopamine is being released into this brain region. So it looks something like this. place. Sorry, it's a little fuzzy. Here, the mouse is just going to be walking around, and then the sensor will change in fluorescence, and that signal will be sent through that cable there up to a computer, and that's what you see on the red line. So dopamine increases and decreases in real time as the animal's behaving. So what happens if we do our experiment with the tone and the foot shock? So here you see the auditory cue turning on and off. That's the gray box. That's a little hard to see, I apologize. And then the foot shock is the lightning bolt. So the graph indicates when there's an increase in dopamine release in the hippocampus. So as you can see, when the tone turns on, there's a little dopamine that gets released, so the brain is paying attention to it. Same thing when it turns off, but not nearly as much as when the animal thinks it's being attacked. Then you get a huge increase in dopamine release in the hippocampus. So that's nice, but is that actually important for memory in this case? So to test that, what we did was to lesion or damage the locus coeruleus that releases dopamine into the hippocampus. And then we measured dopamine again during learning. And as you can see here with the orange line, if you get rid of the locus coeruleus, you get a huge decrease in dopamine release in the hippocampus during learning. You still get a little bit, but most of it is gone. So, does that affect the animal's ability to form a long-term memory and remember this cue? And it does. So, I don't have to describe freezing. That's good. So, this is the fear behavior we use to look at memory. And as you can see in the controls, during the baseline period, they don't freeze very much, but as soon as the tone turns on, they go into a very strong freeze, which indicates to us that they're scared and they remember this auditory cue. And that memory is significantly reduced when we reduce dopamine release in the hippocampus. Okay? So, but, it's science. We need more than just one piece of evidence. So I'm only going to show you a few, but we did a whole bunch of experiments. Here we blocked dopamine receptors with a drug called the dopamine antagonist. So we infused this drug into the hippocampus during learning, then we train them, and then the next day we bring them back to see if they remember. And here you can see that the dopamine antagonist almost completely prevents learning about the auditory cue. And so what this tells us is that when there is dopamine being released into the hippocampus during this learning event, it has to bind to its receptors and induce plasticity if you want to form a long-term memory and remember this cue for a long period of time. Okay, so 2 examples of knocking down dopamine and impairing memory. Next, we wanted to do something different. We wanted to know if we could take a memory that was initially weak and make it stronger with dopamine. I don't think bodybuilders take dopamine. I think it's something else to get this big. But we're talking about memory, so we know that, so we're going to ask, does dopamine make memory stronger? So this goes back to the beginning of my talk. So remember I told you when you're on a morning commute that's very boring, you wouldn't probably remember it very long. But what if you had that same boring commute and I put a bunch of dopamine into your hippocampus? Would you suddenly now remember it for a long period of time when normally you wouldn't? So that's what we're asking with this experiment. So to do this, we used a tool that Diasnu mentioned earlier, optogenetics. And so we can use this to deliver pulses of light into the brain and activate neurons via fiber optic cable, which you see in the rat's head right there. And so we use something called an opsin. It's a light-sensitive protein, kind of like the opsins in your retina, but it's a little bit different. And when we activate these proteins with blue light, the neurons will fire a bunch of action potentials. And so if we do this in the locus coeruleus, the locus coeruleus will release a bunch of dopamine into the hippocampus. Okay? So what we wanted to do is train the animals with a weak protocol they wouldn't remember very well, and then in one group, stimulate dopamine release in the hippocampus during learning to see if they now remembered. So here's the control group. We used a very weak shock. The animals barely pay attention to it, and you can see they show almost no fear to the tone, so they don't remember the tone very well at all. But if we stimulate the locus coeruleus and then give them this same weak training protocol, now we end up with a really strong memory. So we can turn a weak memory into a strong one by increasing dopamine release in the hippocampus. So to summarize, salient events increase dopamine release in the hippocampus, other parts of the brain too, but in the hippocampus, they enhance episodic memory. Blocking dopamine release or blocking its receptors in the hippocampus impairs long-term memory formation. And if we artificially increase dopamine release during learning, we can turn a weak memory into a strong one. So future directions. This is something we're doing right now. We are imaging the activity of hundreds of neurons in the hippocampus, that's what you see here, through these things called miniaturized microscopes. And so we implant these, we implant a lens above the hippocampus, and the neurons are engineered so what they fire action potentials, they turn green, and that's what you're seeing flashing on the screen, although I don't have the color on there. So those are hippocampal neurons firing as the animal is learning. And we can also see dopamine release at the same time. And what we want to know is, how does dopamine release affect the patterns of activity that we're seeing in the hippocampus, the firing of different cell types, correlated firing in a way that enhances long-term memory. Okay, so the stuff most people care about. What is the relationship between dopamine and human memory? Now, I don't study this, but, and I apologize, that looked way better on my other program. It's a little fuzzy there. But there's a few things that we know. One is that dopamine activity, or the number of dopamine neurons, do decrease with age. And at the same time, as most of us know and have experienced, our memories get worse as we age. And these two things are correlated. So what can we do? Well, there are a few things that we have control over that can help, but these are kind of boring because they're things that we know we should do anyway. These things will increase dopamine levels and enhance memory in aging. Exercise, good sleep, socializing, and good diet. But what about treatments? Diazmi mentioned this a little bit. So in the laboratory, we actually have lots of things we can do to increase dopamine release. So you might have heard of L-Dopa for Parkinson's. We have inhibitors for the dopamine transporter, which means there's more dopamine at the synapse. A lot of those are used for ADHD and conditions like that. And there are many, many more. But there are some very difficult issues to overcome. First, we would like to manipulate dopamine levels in specific brain regions and in specific circuits. We can do that in animals. We can't really do it in people yet. In people, we give them medication. But as I just showed you, dopamine does lots of things, right? And so you have side effects, maybe motor side effects. Dopamine is rewarding in some areas. If it's too rewarding, it becomes addictive. You don't want that either, right? So it's very complicated to try to fix. And so what the NIH has done is to create something called a funding program called the BRAIN Initiative. They did this a few years back. And the point of this initiative is to take these basic science findings that we have in laboratories like mine and get teams of scientists to work together and figure out how do we use this in the brain, which is a difficult problem. So at UC Davis, a number of people have received these grants, and it brings together biomedical engineers, the people have to really figure out these, how do I get a drug into a brain in a specific region? I have neuroscientists, psychologists, all working together to try to figure out how to take basic science and apply it to human health. And so with this kind of funding, the hope is that in the near future, we'll start to see more of these things we know in the lab be applied to human health. And with that, I will end and thank my laboratory and funding sources.
You mentioned neuroplasticity, but you didn't actually go into it. Is it believed that neuroplasticity is how memories are formed?
Yeah, so the sort of, the way that we envision it is that there are groups of neurons that become more, that will represent some event in the world. And those groups of neurons during learning will strengthen their connections with one another. And so now you have a group of interconnected neurons that represent different aspects of an experience. And so now I can think of that whole experience if I can reactivate that whole ensemble of neurons. So in order to do that, they have to strengthen their connections. There's a number of ways they can do that. Karen Zito, who was here earlier, can tell you all about changes in spine growth and synaptic strength. Kim can tell you all about changes in synaptic strength. And we think that those allow those groups to wire themselves up, and that's what memory is.
So in treating psychotic disorders, we use, or at least 30 years ago, very commonly used very potent dopamine blockers. Would that affect the formation of long-term memory in those patients?
Yeah, I mean, that's a good question. I mean, I know there were lots of side effects, right, that were undesirable. So it's complicated about what effect it would have on memory because those drugs go everywhere. So, and there's multiple dopamine receptors. There's D1, there's D2. Some of those receptors are on excitatory neurons. Some of them are on inhibitory neurons. So what effect would giving a drug that blocks dopamine receptors all over the brain have? It would probably impair memory, but it would probably do a lot of other stuff too, just because it's sort of a blanket treatment and you're not doing a fine-tuned sort of thing. So I would guess it impaired memory, but I don't know 100%.
I don't know if this has been looked at, but dreams, you know, you presumably are having a dream based on a past memory. You have a dream, sometimes you remember your dreams, most of the time you don't. Is that neurotransmitter based as well? You know, it's a dream,
Yeah.
a very vivid dream, and transmitters are released.
In fact, in that imaging study, well, you saw all the neurons firing. So we have a protocol, so we image first, then we train, then we put them in a nice little area with a blanket, and they go to sleep, and then we keep imaging while they're sleeping to see the neurons being reactivated. And so, yes, so typically what happens is, so it's interesting, so the hippocampus will sort of replay events that just happened, and they will do that immediately after the event, but they'll also do it right after you go to sleep, so slow wave sleep. So usually if somebody wakes you up shortly after you've gone to bed and they ask you what you were thinking about, you'll be thinking about the day. And that's because the hippocampus is kind of replaying what just happened. When you get to REM sleep and somebody wakes you up, then it's just weird. No one knows you're on a mountain with a unicorn and no one understands what that meant. But early in sleep, during slow wave sleep, the hippocampus replays, and people have shown that that's actually important for strengthening the memory as well, to keep replaying it.
You showed two graphs on there that showed the dopamine reaction and memory declining with age, but there were no units on the vertical axis.
Yeah. So there's several numbers you can use.
So what are the units on the vertical axis?
So you can look at volume of dopaminergic areas, so areas that have dopaminergic neurons. Unfortunately, they shrink with aging, those areas. You can look at dopamine receptor number, and the number of dopamine receptors on neurons also decreases with aging. So that figure there was actually volume, but you can also see the same thing with receptor number.
What about the memory one, the second?
Oh, memory. Oh, yeah. So with episodic memories, typically in those types of studies, that'll be done kind of, if you're going to actually look at episodes from a person's life, you'll have them go through experiences and then bring them back a day later, a week later, a month later to see how well they remember. And typically your memories fade much more quickly when you are older. Now, interestingly,
I don't think your graph tells me that though, because you don't tell me the conditions, you don't show the units.
Sure.
I don't, it sounds like it could be confirmation bias or just conventional thinking. I think you have to demonstrate with the units and the test conditions what it is before you can make that statement.
Oh
from
yeah,
that
sure,
graph.
sure. Within the papers, there are units. but they can be complicated to explain, especially in a general audience. So, but yes, for sure in those studies, they have metrics, quantification of episodic memory, how long it's remembered, how many details you remember, and those things fade as we get older. But interestingly, I was talking with Charn Ranganeth.
But it makes a difference whether it's an absolute number or a percentage. As you get older, you have more experiences. You have more episodes in your life. So, you know, if I'm a 30-year-old and I have 3/8 the experiences of an 80-year-old, the fact that I can remember 3 out of 8 isn't such a great, response compared to when I'm 80 and I can remember 5 out of 8, but you might conclude,
yeah, that's a
sorry,
great
3
point.
out of 3 versus 5 out of 8, I'd give it to the 5.
Yeah, that's a great point. So,
But you, as
so
a
it's
percentage,
true.
you say, hey, your memory declined.
So it's true that the number one cause of forgetting is interference. Interference, as you said. So the more experiences you have after you've learned something, the more it's likely to interfere with that event. So that is true. In the laboratory, these animals don't have many memories at all, which is sometimes why they can actually remember them for a long period of time. But in those studies that they'll do, looking at memories over aging, a lot of times what they'll do is use cues or examples or things that you have to remember that you've never seen before. You have no experience. Often they're even like made up symbols or words. And so if an individual who's older goes through one of those tests, they're more likely to forget some of that information than someone that's younger. A little harder to explain that in terms of experience, although you could probably think of maybe some ways, but it does seem that we forget more quickly as we age.
But those tests are short-term semantic memory. They're not episodic.
You can do long-term memory tests with those as well.
Well,
not
anyway.
just short-term. Yeah.
I think that there has to be more rigor before you draw that conclusion. It's more like a conventional wisdom pronouncement than
Yeah, I mean,
something
it seems like
supported by the
it's
data.
also just not just episodic memory, but other forms of memory decline with aging as well, working memory, things of that nature. Yeah, sure. I can give you the study afterwards if you want to take a closer look at it.
Hi, your presentation was
Yeah.
super interesting. Thank you. I wanted to ask, so if dopamine reinforces long-term memory, if I pretend to be happy when I'm studying, am I going to have the material in my brain for longer?
No, this is something that I was going to say just a second ago, which is, so I was talking to Charn Ranganeth, who's our New York Times best-selling memory expert now, and we were talking about this, and my mom would call me and she'd say, "Oh, your dad, he's I'm a little bit worried about his memory, he's forgetting," and so I'll call him, and he's a sporto. And so in Iowa, we have one team, the Iowa Hawkeyes. There's no professional sports. So I'll start asking him about that. He remembers everything. He remembers the game, the quarters, the plays. Why did we run it on that one instead of throwing it on that one? What is the coach doing? That defensive call was terrible. He can tell you everything about Caitlin Clark, who's the most famous person in Iowa right now. And it's because he's very interested and curious about it. And so I talked to Charan, and he said, oh, that's amazing, because they were doing some studies where the subjects could choose either to learn some fact that they thought was interesting or some fact that was just given to them. And when they were given information to learn that they were interested in, it activated dopaminergic regions and they remembered it much better. So what Charan was saying is that oftentimes, right, if we're very interested and engaged by something, which you've probably seen in school all the time, you remember it much better. And when it's a subject you don't like, it can be harder to remember. So I'm not certain if that's related to dopamine, but it's possible.
I have a biography question. When you first started your talk, you mentioned Lashley and his feeling of complete failure at discovering what he sought to. And I noticed that he died at a very young age. He was 35, and I'm wondering if that despair had anything to do with his death.
No, sorry. Those were the years he was studying memory, not the years of his life.
Well, that wasn't his lifetime.
Sorry.
I'm glad to hear that.
Looked a lot older than 30 in the picture. But actually, it's a little bit unfair, I think, because that's how Lashley is remembered, ironically. But he actually did a lot of other really interesting things that pushed neuroscience forward, but that's the thing he's remembered for.
Last question.
Last question. Very briefly, thank you. There's two quick comments. As we age, I've noticed that retrieval is the main problem.
That what?
Retrieving.
Oh, retrieval.
Retrieval is the main problem. And the other thing is that when I was at university, I remember it was common knowledge that you should study late in the evening and then go to sleep, and the next day you remembered everything. But actually, my question is this. How is memory affected in the digital world? Because, I mean, that would be an interesting thing to study, maybe even what happens in the brain. Because
Yeah, that's a good question. I mean, I don't know of, studies that have correlated. Are you talking about like social media and games and what impacts they would have on? Oh, I see. Yeah, I mean, it seems. I mean, there are definitely some things that we know enhance memory. I give this paper to my students every year, and none of them listen to me. It's a paper that shows that it compares students who take notes by hand versus typing, and they remember much better if they write it down. And so I show it to them, I post it on Canvas, and then they all get on their computers and right? So, but there, in that particular case, right, it's usually kind of like a level of encoding thing. You're putting effort into it, you're thinking about it, you're drawing a graph, as opposed to just looking at a slide and passively getting the information. The other thing, it's a little bit different, but it's related to retrieval. And so, in fact, they found that one of the best things that you can do to remember is not to restudy, but force yourself to retrieve. And so if you learn something and you wait a period of time and then you force yourself to retrieve it, you get a huge bump in retention, even more than restudying. Because most of the time when you restudy, you just kind of read it again. But if you force yourself to retrieve it, right, they call this thing retrieval strength, you can actually bump it up and then you remember even better.
Thank you so much, Dr. Wiltgen. We're out of time. Thank you.